This is episode 321 of the Stem Cell Podcast, Decoding Blood Stem Cells with Dr. Vijay Sankaran. Hello, everybody. This is Dr. Daylon James. With you in a minute will be Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The pod is brought to you by STEMCELL Technologies, a global biotech supporting life science, research, and fostering communication and collaboration in science. If you're enjoying this, listen, rate us, leave a review. We're always looking for feedback on how we can improve and for suggestions on some new guests that we haven't had on the show before. Maybe some that we have had that we should bring back. Come on, send a message. Today, for our guest, we have Dr. Vijay Sankaran from Harvard Medical School. He's on the podcast to talk about his research on how human genetics is transforming our understanding of blood development and driving new therapies for blood disorders. We've also got our usual roundup, although a bit abbreviated today. My apologies. Our usual roundup of recent highlights and stem cell news. That's coming up in just a sec. But first, a quick heads up. If you're in stem cell science, you need to be at ISSCR 2026. We're talking an unreal lineup here. Fen Zhang, Aviv Regev, Shinya Yamanaka, Peter Zanstra. And that's just the beginning. Catch the latest breakthroughs live July 8th through the 11th in Montreal. Go to isscr2026.org and lock in your spot. Lock it in, guys. There'll be spots for you if you get in there soon, but you don't know. You don't know. Montreal's not a huge city. They might fill up. Lock it in. Moving on to the roundup. Today, I've got a bit of disappointing news, which is that due to a scheduling mishap, it's just me flying solo. Arun can't be with us, but fear not, you Arun fans, which makes up the lion's share of our audience for sure. He'll be back in the second half of the show, and he left with me a little gem, a little piece of his heart, from his heart to yours, that I'm going to close out the roundup with. And, bad news for the Sharma fans, but good news for the James fans. Talking about you, Mom. We got an uninterrupted monologue here, a mind-bending synthesis of the science in our midst. And we're starting with a piece that is kind of on point, apropos to our guest, who's really focused on how hematopoiesis goes awry in disease. I got a story from my neck of the woods here at MSK, Danwei Huangfu, who's in the developmental biology program there, but also at the Rubenstein Center for Pancreatic Cancer Research, because it turns out there's a lot of alignment and symmetry to the mechanistic processes that underlie development and neoplasia, right? And here we're talking, as you might imagine, about pancreatic cancer, which it's a bad one. We all know that. But part of the reasons there is because the diagnosis of pancreatic cancer, which is specifically in most cases pancreatic ductal or adenocarcinoma, it occurs at really advanced stages, right? So the prognoses for pancreatic cancer is the grimmest, and among the grimmest, that and glio, it's the four horsemen, two others probably in there, but it's among the worst. And because perhaps in part that you catch it so late, the mechanisms that drive the malignant transformation at the outset, when the neoplasia arises in the pancreas, it remains really poorly understood. But what few samples you have clinically, what little understanding, I mean, significant understanding, but we could know a lot more. The understanding we do have of that pancreatic intraepithelial neoplasia is in these clinical samples, these like precancerous neoplasias, in the epithelial lining of the pancreas that's like a common precursor to full-blown adenocarcinoma in the pancreas. Um, and what we know from that early precursor is that even at that point, at that relatively early point, right on the cusp of forming cancer, the four most altered genes in the full-blown cancer, which are KRAS, CDKN2A, P53, and SMAD4, they're all already manifest, those mutations in those early precancerous stages, right? Um, and because they all co-migrate and the how pervasive the alterations are in those four genes, it suggests that there's a cooperative uh, element there that drives neoplastic transformation before you even get the development of the malignant uh, adenocarcinoma, right? And there are genetic mouse models, of course, that enable you to recapitulate those mutations relatively straightforwardly. So you use these genetic engineered mice, and it supports a lot of that mechanistic insight into the links between those four genes. But there's some discrepancies between the mechanisms of cancer arising in the mouse models and the clinical observations that you see in human patients, right? So, of course, we need these more faithful human cell-based models for pancreatic cancer in order to unlock particularly these early stages, like what's going on there. So that's where Dan Wang and her group and a lot of collaborators, also from my institute, Weill Cornell and elsewhere, they went in on this with I think a really unique approach, which was capitalizing on the symmetry between pathological neoplasia in adult mature tissues and these developmental events. They used pluripotent stem cell derived pancreatic progenitor organoids as a kind of model of this precancerous pancreatic ductal adenocarcinoma transformation, right? And by kind of modeling this, what has been observed clinically, this inflammatory, and in mice, really not clinically in mice, that if you dump this inflammatory insult on the pancreatic epithelium, you can get this kind of oncogenic transformation. So they capitalize on that kind of basic cellular process to create this model using that's based in human ES cells, and they validate it. So the dev cell paper. Um, they validate the model against these clinical data sets, showing that it recapitulates this progressive, um, both the initiation and progression of pancreatic cancer, and then going mechanistically, and they show that the loss of CDKN2A is essential to the transformation when combined with these fundamental KRAS and P53 mutations, while the SMAD4, which is the fourth gene that's been always observed in the context of pancreatic cancer, that is more about tumor progression. It's not essential for the actual initial transformation, right? Then they look in, they show that there's this epigenetic link uh, that uh, it uh, favors this oncogenic program at the expense of the developmental lineage specific pancreatic lineage specific program, right? So it looks like there's these competing agendas here that are modulated epigenetically, and then they show that that is a TET mediated um, phenomenon. So TET mediates suppression of ERK signaling, and then the epigenetic consequences thereof, this hypermethylation of these pancreatic specific transcription factors. So a lot of links in the chain here mechanistically, showing that ultimately that favors this oncogenic program at the expense of the pancreatic specific lineage progression. So like all in all, this is a beautiful study, I think, that hacks the ES-based system to to generate a kind of counterintuitive, maybe, model of pancreatic cancer, which is a really necessary and going to be instrumental, I think, revolutionary potential tool in understanding and addressing the grave prognoses in this disease. Um, But then it goes actually into this kind of fundamental transformational node, and elucidate some of the mechanistic links there, and with this, I think, interesting conceptual understanding, insight of that they are losing their, which makes sense, obviously, but to show it on a mechanistic level that these things that are meant to be developing in within the pancreatic lineage get hijacked and just go a different way, which totally makes sense, obviously. And then TET as like a maybe potentially therapeutic strategy for suppressing this. Like if you were to extend that to patients, maybe there's a way where you could identify these patients earlier using this model system to identify biomarkers that are perhaps detectable. And then on those at-risk patients, you could even mitigate their risk by suppressing this maybe perhaps mainline TET-mediated mechanism of transformation. So, hey, it's got a lot in it. Very developmental, but cancer elements and, basic with the epigenetics and developmental, obviously, with the lineage progression. I love a story that has, I can't say it has it all, but it has a lot. Um, and also, just from a 10,000-foot view, I think really underscores the value of the developmental biology program in a major world-class cancer center like MSK, which a lot of people ask, like, why the stem cell program or how they have such a strong, I mean, really famous almost stem cell program there. And it's because there is like a hand in glove relationship from a therapeutic standpoint, both with cell based therapy, but also basic understanding of cancer progression. So this story really does have it all. Well done, Dunway. Uh, I wish Arun had something to say about this, but he doesn't. So I will continue on with the exhausting monologue. But I'm pretty excited. I hope you guys are too. I'm going to stay with this disease angle, and this will be a quickie. So we're gonna get through this, guys. And there's so much more science out there that I didn't cover. Just think about that. I'm trying to spare you, but not from this. This is a great extension of I think the story I just talked about, which is coming from a pluripotent stem cell foundation for disease modeling. Here is much more direct cancer stem cell story and patient specific but technical. And I think the reason why I want to highlight this is because I think this is where we are. We're in all the stories today. We're going. You'll see with the next one. We're like really at the bleeding edge, pushing the limits with proof of concept modeling. We've got the real, soup to nuts, boots on the ground modeling of pancreatic cancer. In that case, it's getting candidate therapies. And then here, this story, it's about the tool set, like truly the consumables that we need to generate nowadays in order to capitalize on all the scientific potential of our tool set, right? So let me elaborate. We're talking about cancer stem cells here. Of course, the thing with cancer stem cells, it's in the name, right? These are the cells that really drive the initiation, the progression, the resistance, the recurrence, the spread, metastasis of cancer. It all comes from the stem cell, just like in development. And perhaps not surprisingly, the cancer drugs that have been approved by the FDA, uh, among over 100 of which that were studied recently, the most effective uh, drugs that are associated with the best clinical or the most improved clinical outcomes are the ones that target cancer stem cells, right? Not surprisingly. So the idea, and this, I think, the idea has been percolating for some time, clearly, but I think now we've reached a point where technically, and the will is there, I think, to be going wholesale uh, generating patient-derived cancer stem cells for drug testing, like on a study basis. That's already there. But I think from, I'm not the only one. I think from like a clinical precision oncology standpoint, like standard of care standpoint, the idea of using cancer stem cells for drug testing in perhaps all comers amongst cancer patients or maybe even other malignancies. That's an idea, right? That I think is maturing. Um, but there's still a lot of challenges, right? The isolation of these functional cancer stem cells, it's technically difficult, right? There's a lot of inconsistencies. Uh, it's how well they mirror the in vivo uh, biology in some cases can be highly variable. And really, it's about the scaling up, right? If we're going to do this wholesale for each patient without significant expertise amongst all the operators, it's time consuming, right? And it's costly, and you do need that expertise. So this is a technical report in Nature Cell Biology from Zhangjuan Zhu, Hao Zhu, and Guangli Shuo. Doing my best with the pronunciations. Apologies. If I got a few of those off, they're all from Chuchao, China, the Chinese Academy of Sciences there. And they, together with their groups, made this plate. They made this microwell cell chip culture platform to enable quick and high throughput generation of these micro tumor spheres uniformly. And here they focused on breast cancer specimens from biopsies, right? So real, not even like experimental in the lab. The whole point here is that this is something that could be scaled out in every clinic with perhaps some modifications, of course. And they show that they had functional cancer stem cell enrichment and personalized drug testing from those cultures within just over a week, within 8 days of the biopsy, right? So we're talking certainly on the timescales where you can use this in advance of making a definitive clinical judgment for the treatment plan of that patient. And then they use single cell limiting dilution assay to validate this. And this is a key here in terms of proof of concept. They showed that these tumor spheres predicted the clinical outcomes in a cohort here of 16 patients that were the preliminary run breast cancer patients here with 93.8% accuracy. I'm guessing that's 15 out of 16, right? So pretty high rate, which, given the time scale of 8 days, really underlines how ready for prime time approaches like these are. And as I said, like kind of as a bridge from the previous study, which is like work that's done in labs, and the study I'm about to talk to, which is work that's done in one lab. Uh, this is something that could be in the clinic tomorrow. I mean, it's plasticware and existing material that is in wide clinical use and practice. So, including methods that are in pretty wide practice nowadays in many centers. So, yeah, I think we're ready here to see a lot of reports like this. I like that Nature Cell Bio will throw in a technical report like this because maybe it's something that wouldn't be showcased in Nature Medicine. I don't think so, or Nature Methods maybe because it's not highfalutin enough. But this is a bridge and something that I think a lot of groups are going to see and be like, why am I not using this with my patients? So they're going to be making some money off of that. Maybe they're giving it away free. I wouldn't be surprised. Anyway, moving on as promised, a little taste of some Arun love here. And I can't approximate his scientific acumen or voice. That goes without saying. But his fascination, interest, and investment, maybe not the total scale of the investment, but the interest and fascination for sure with the heart, and particularly the human pluripotent stem cell based systems that try and model heart development and pathophysiology. Love it. Love it. And that's what I'm gonna finish on here. It's a little mini roundup here, only three, but it's for your sanity, because four would be too much, wouldn't it? Um, we'll 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 balance the scales here with Arun. He can take the lead with VJ. Uh, moving into Arun's milkshake here. This is a story about the heart, as I said, but more specifically, we're talking about the sinoatrial node, right, which is the primary pacemaker in the human heart that provides the electrical impulses that initiate each heartbeat, every single one, right? And it lives right beneath the epicardium at the junction of the superior vena cava and the right atrium. So it's this little node, right? And the complex, right? Because it is this 3D cluster, the pacemaker conduction complex, it is comprised of a heterogeneous mix of pacemaker cell subtypes, right? Including these head, tail, transitional populations, right? And I mean, come on, it's the heart and it's pluripotent stem cells. It was the first thing that anyone ever probably went gaga over because you saw it beaten right there in the dish. So they've done it all in the heart, but clearly not at all yet because it's like a complicated machine, right? And there's a lot of challenges of getting all those pieces to work together, right? We're not talking Legos here, guys. So from the human pluripotent stem cell models and diffs, we've generated the community, not including me, frankly. I haven't done enough. But the community at large has generated these sinoatrial node-like pacemaker cells, but they generally lack the 3D organization and the heterogeneity, and specifically here, and this is a key factor, the autonomic innervation, right? The innervation. When we talk about impulse, there's always going to be a nerve somewhere around there. Um, and the cardiac organoids, right, on the other side, or on one side there, on maybe like one arc length of the circle of many different things that have been made in the heart field from human pluripotent stem cells, there's these cardiac organoids that have tremendous potential to model development, disease, kind of, the myocyte diseases in particular, and many aspects. Um, but they don't capture the regional specialization, and again, that neuro cardiac interaction, at least not sufficiently to model the sinoatrial node. So here, in a story that was published in Cell Stem Cell from An Zhang, from the Chinese Academy of Sciences in Shanghai, they went like Rambo. I don't know. I'm picturing one of these days. This is I'm aging myself with this reference, but there's going to be like a Voltron like organoid where they pull in the bang, 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 four joints, and then the head, and the organoid is going to walk out of the plate. I hope I'm alive to see the Voltron organoid. But here they're taking a swing at it, right, with a tri-assembloid, which sounds like it came right out of the Voltron cartoon. I wish I had seen that episode. They should have made it. But here they take a threeplex approach with the sinoatrial organoids, right? Bang. Then they add to that these cardiac ganglionated plexus organoids, bang, bang. And then add to that some garden variety atrial like cardiac organoids, right? So with that triplex, they model pacemaker to atrial conduction, right? And it recapitulates the molecular, structural, electrophysiological features of that human pacemaker activity. And this they take it to the hill here. It does that model is sufficient to allow interrogation of the neural input, right? The neural control over the sinoatrial node, and specifically here looking at conduction dysfunction, right? So like this, we're talking about not just like cardiac genetic disease, whatever. which is all very intense and amazing studies. Timothy syndrome. Is that one of them? I don't know. There's a lot of them. Arun could tell you long list of disease-specific cardiac models. But like the elephant in the room here is that we got to be modeling the big killer, right? The conduction dysfunction. Isn't that the big killer? Maybe I'm a bit off there. Arun's gonna scold me on me throwing shade here, but that's not what I'm trying to do here. I'm just saying this is so dope because they model the conduction function and then going to bona fides and spatial transcriptomics on human sinoatrial node tissue. They combo that ground truth up with the assembloid-based functional analysis, and they get a little mechanism in there. They show that there's a kind of signaling relationship between this prosaposin and GPR37 that is a key to pacemaker maturation. So just a little kernel, right? Just like a little nibble off the model to show how you can use this system to kind of unlock and elucidate the crosstalk that's going on in this complex machine to generate a mature unit. And that's what we talk about a lot, maturation from like a cell intrinsic level. But I think a big part of the problem is that we got to work on maturation of the whole joint. And that's what we're talking about here. How can we model the inter uh, connectivity and the truth. This is what the assemblages are all about. But here with the tri-assembloid, all they need is the head. And I think we got somebody write that up. Anyway, the complexity of the organoids. If Arun were here right now, we'd be chatting about how we're going next level. And this isn't even to speak to how you can imagine integrating, endothelial, I don't know, immune. We're going to be talking about the immune cells and heart repair in a couple of weeks with Philippa Simoes. So I don't know. We got to unpack this a little bit with her. But first, and this is the last roundup story and near the end of my screed. But I hope it was a stimulating one. Because we went all over the map, starting with these disease models, and I think we're gonna circle back to that with our guest in just a minute. Vijay Sankaran tries to understand how hematopoiesis goes right and wrong. But first, we have a quick message from STEMCELL Technologies. Do you work with human pluripotent stem cells and want to strengthen reproducibility in your research? Learn how with applying the ISSCR standards for research, a free on-demand training course offered by STEMCELL Technologies and the International Society for Stem Cell Research. This self-paced online course provides practical guidance on best practices for cell characterization, pluripotency assessment, genomic integrity testing, and reporting. To learn more and register, visit stemcell.com/ISSCR-standards-course. All right, guys. Today on the show, we have a very special guest. There are always very special guests, brilliant leaders in our field. But today we have a real fluent communicator who you love going to his talks because he really gives you a peek behind the biological curtain. In my near and dear erstwhile focus, hematopoiesis, Dr. Vijay Sankaran, who is a medical professor at Harvard Medical School, investigator at the Howard Hughes Medical Institute, attending physician at Dana-Farber Boston Children's Cancer and Blood Disorder Center, also associate member of the Broad Institute. And a lot of our listeners know the work, but just a brief snapshot. The Sankaran Laboratory uses human genetics and many other tools to study hematopoiesis and how this process goes awry in human disease. The group is particularly interested in gaining molecular insights into how hematopoietic stem and progenitor cells are able to effectively produce mature blood and immune cells. The lab is also interested in identifying opportunities to use the insights we gain from our studies to develop improved therapies for the variety of diseases. We just talked about one of those studies that we're going to showcase a bit on the show today. We just talked about that last episode. We'll come around to that, but thank you, Vijay, so much for joining us on today's show.
Well, fantastic to be here. Thank you both for having me, and really looking forward to this conversation.
Yeah, likewise. So, like Dylan said, your lab is at this really fascinating intersection of human genetics, hematopoietic stem cell biology, translational medicine, and of course, you're at a perfect place to support all that at the Heart Stem Cell Institute. Could you start by giving us an overview, kind of what your group is focused on these days, sort of in your own words, and how do you think about connecting these fundamental discoveries in blood development to therapies for patients with different hematologic diseases?
Yeah, no, thank you for that question. And maybe I'll just go back a little bit historically and give you a sense of kind of why we're doing what we're doing. So, I have not made it very far in my career. I literally was a graduate student down the hall working with some folks you've known. I was a benchmate of Sean Wu, who Arun, I know you have worked with, and was here as a graduate student and trying to figure out what I would end up doing. And I bring this up partially because this was in the early 2000s. At the time, the workflow was really, how do we knock out genes in mouse models, make tissue selective deletions or mutations, and then examine the phenotype and really do either transplantation in the context of hematopoietic stem cells or in other projects that were happening in the lab at the same time. And I was a PhD student with Stu Orkin at the time. There was work on pluripotent stem cells, there was work on cardiac stem cells. So there's a lot of stuff that was kind of happening around that time, that was really exciting. But one thing that I really wanted to do was to try to say, could we address some problems that hadn't been addressed before? And it turned out part of the reason we initially got into human genetics was I really wanted to understand this fetal to adult hemoglobin switch. I was determined to do this in part because of patients that I met. And that really inspired us to try to pursue this avenue. We kept failing. We're trying all these systems like, taking humanized mice, examining the switch in those settings, doing at the time, siRNA screens, which in retrospect seems antiquated, all these sorts of things. But the important lesson I took away was the one thing that helped us advance was really thanks to work that had followed up on the Human Genome Project that had been published in 2003, but all the work on HapMap and so forth led really to the possibility that we could use genome-wide association studies to look at associations. And that really led us to a key regulator of fetal hemoglobin, BCL11A, now an important therapeutic target from a variety of cellular therapy approaches, whether from Exocel or Casgevy, which targets the enhancer of BCL11A. Other approaches are using lentiviral approaches to suppress BCL11A, or even most recently, there's been work from a couple of companies, including Beam Therapeutics and Correct Sequencing Therapeutics, to perform base editing in hematopoietic stem cells to target the binding site of BCL11A. But I tell you all of that because that's really what our North Star in the lab is, which is how can we use human genetics to maybe go beyond what has traditionally been happening or that's complementary to what's been happening in many aspects of stem cell biology, and actually bring this to bear to understand more about how we can better understand basic aspects of stem cells, but ultimately, hopefully, bring that back to so many of our patients.
Wow, I'm glad that you led with that because it really explains a lot. Because when I think of your work, I think of just a diversity of questions, diseases,
Mhm.
tool sets, but it's all within this framework that the Human Genome Project served as a launching pad, right, for this endless sea of information and insight that lies sometimes hidden or hidden in plain sight within the sequence there. And that, I mean, you started at the beginning, but I'm going right into recent history, which we touched upon in the jump about this paper you just had in Cell,
Yeah.
covered in last week's episode. And it's a good segue there because I loved the story, start to finish. But what I really loved was that serendipitous element, maybe. I mean, you could call it serendipity. You called it in the actual intro. It was a serendipitous finding. That element of the narrative really touches on that, adventurous notion of all of us as scientists of seeing what no one has seen before, and sometimes right there, this novel mechanism hiding in plain sight. And perhaps, as Arun alluded to, the most satisfying acronym that has ever been devised, HOTSCRAMBL, which, I said at the time, sometimes these acronyms leave you scratching their head about either how they came up with the letters or what any of it means. But this one is right on. I hope you trademark that thing immediately. But tell us more about that story and maybe how, as you finished there, how you can bring all this information to patients. Like, how do you, because that was what ultimately seduced me, was that it had this binary element of being both insight about, normal function, but something that was targetable in the context of pathology in AML. So tell us about that story a little bit more, and how you envision the translational potential.
Yeah, no, absolutely. So, that was really somewhat of a serendipitous or, observation. One of the things we've been really focused on in our group in the last few years has been to try to understand genetic variation that either puts individuals at risk of getting different blood cancers, particularly some of the myeloid blood cancers like myeloproliferative neoplasms, myelodysplastic syndromes, or acute myeloid leukemia, all of which are thought to arise from mutations that accumulate within stem cells, although we know that there are many individuals who harbor these mutations in otherwise healthy stem cells, in a condition called clonal hematopoiesis. So we've been very motivated to understand what factors modify once you've gotten a mutation, the likelihood of progressing to a blood cancer, or what factors either increase or decrease the risk of acquiring one of these mutations in a stem cell, and acquiring clonal hematopoiesis. And so that's sort of been the thread of work that's led us in this direction. And at first, much of our focus was on looking at risk factors for these diseases, trying to understand some of the biology there. But what's happened over the last couple of years that I think has been very interesting is just as we've been able to find risk factors, and it's important to understand why individuals are at risk, what's becoming even more evident is there are a number of genetic variants that actually protect individuals from acquiring either clonal hematopoiesis or from acquiring one of these blood cancers. And these are what we've sometimes termed resilience variants. We've worked in a few instances on some other resilience variants. For instance, we identified a variant that affected a key stem cell RNA binding protein, MSI2. And this was in a regulatory element of MSI2 that was within blood stem cells and therefore affecting its expression there. And this actually, by decreasing the activity or decreasing the expression of MSI2, actually protected blood stem cells from acquiring either clonal hematopoiesis mutations or from progressing once they'd acquired a mutation to blood cancers. But I bring that up because I think that's an example where there was already so much well worked out biology that folks had done. A number of folks had contributed in our field: Kristen Hope, Michael Karas, Gary Gilliland. So many labs had really contributed to this, that many of whom I'm not even able to name as a kind of full list, but others, including George Daly and Tanish Dareya and so forth, had really contributed to our understanding of this axis. And so there we had all this biology we had known about. In this particular instance, we actually identified a variant in the HOXA gene locus. And my first reaction was, well, this is going to be somewhat boring because, people have studied the HOXA locus for decades. It's so well understood, so well conserved across different species and so forth. And of course, there's been a well worked out role of the HOXA locus in regulating hematopoietic stem cells, but also in getting activated to to basically enable stem cell programs to be expressed in leukemias and so forth. And that's really thought to be the basis by which some fusions like the MLL fusion proteins actually act and cause leukemia-like features. So I bring all of that up because our first instinct was to say, okay, there's a variant. What is it doing? And much to our surprise, as we actually started to look at the genetic data, we really homed in on a single variant, but it was within a transcript that had just been poorly annotated in the genome, wasn't well studied. And it turns out, and it's a little bit of the retrospect looking back, but this was a transcript that was actually unique to the human HOXA cluster. So this is not expressed in mouse hematopoietic stem cells. It's not expressed across other species, but really seemed to be very human specific. And so as we started to look at this, it turned out this was actually a long non-coding RNA, hadn't been well characterized. And, really we ended up naming it HOTSCRAMBL in part because this was the standard nomenclature. So Howard Chang's lab had started this kind of habit of naming HOXA antisense long non-coding RNAs as hot. They started with hot air and then hot tip. And that was really to name them HOXA locus opposite strand transcript. And then, it was clear to us from some of the early studies we had done, which we can talk more about, that this was critical as a stem cell regulator. So we ended up having that SCR, and then from there it sort of ended up kind of taking a life of its own. So that's sort of how it got the name, and really how we kind of stumbled into it.
What an amazing story. It's funny how things work out, right? Yeah, that's quite a journey. And kind of similarly, your work on fetal hemoglobin regulation had a sort of similar journey. Like, it laid the foundation for some of the first approved CRISPR-based therapy, as we know. You mentioned Casgevy, for sickle cell disease. But looking back, what do you think were some of the key conceptual breakthroughs that allowed that, the field to move from understanding the basic biology, like what you're alluding to, understanding hemoglobin switching biology to actually developing curative therapies? So take us back.
Yeah, well, I think, it's been amazing to see where the field has gone. It was clear to us, although hard to envision, that once we had uncovered the role of BCL11A as being important for this fetal to adult hemoglobin switch and doing some of the earliest knockouts in mice where we could show that this actually delayed the fetal to adult hemoglobin switch in humanized mouse models, that this was a key regulator. Then, of course, the question became, how do you target this? And I think this was a good example where thinking broadly was important, because it actually turned out there were a number of companies that were trying to target BCL11A for therapeutic purposes. In fact, one of the first was Sangamo Biosciences. They had zinc finger-based genome editors. They were attempting to target it. There have been other companies attempting to target the locus. And really, I think what enabled a lot of those advances was, first, there was a lot of work to identify, well, what were the key regulatory elements? What could, be edited effectively? There needed to be advances in the availability of tools. In fact, just as a funny aside, when we published our initial paper in 2008, describing BCL11A as being a key regulator of the fetal to adult hemoglobin switch, on the next page, right after our paper, was a paper I initially ignored that was from Luciano Marafini and Eric Sontheimer, that described this defense system in bacteria. And I, of course, ignored it, but this was, one of the first papers describing CRISPR as a defense system in bacteria. It was sharing like a page with our article in Science in 2008. And I would have ignored it. But I think thankfully, there had been lots of folks, including Jennifer Doudna, Emmanuel Charpentier, others, who had really said, well, you know what? Let's co-opt this tool for therapeutic purposes. So it's in retrospect, it all makes sense and it all comes together, but it also, I think, really emphasizes the general lesson, I would say, that we probably need to always think more broadly about what's happening. I mean, I think it's one of the values of your podcast and others where you're covering a field, but thinking about what are the adjacencies, what are the opportunities. And I think that that's an example where, it was clear that this was a key regulator, this being BCL11A, a key regulator of fetal hemoglobin. But yet, there was such an opportunity that was untapped in many ways that, really required a number of years, some advances to enable us to really go further and make more of that. And so, that's kind of a long-winded answer to your question, but I do think it was a little bit of serendipity waiting over time. I think the other thing, though, that's happening now is I think this really emphasizes the need for fundamental biology. In some ways, what's happening is, you had the first generation, I would say, of Casgevy, which has been very successful, but there's other opportunities by better understanding how BCL11A is acting to silence the gamma globin genes. Now there are emerging base editing approaches. Maybe these will work better. And of course, for all these therapies, there's still lots of challenges that we can talk about. So I think that there's lots to still be done, but also I think it really probably emphasizes in my mind just the need to think broadly about what's happening in the field, where might there be opportunities that we can translate to another field, and so forth.
I love that answer because at both, I mean, on like a micro scale with you sharing a page with Marafini and also on the macro, it's a story, and all science really is about convergence, right? These seemingly disparate ideas that you braid together into innovation that has real impact. And I want to drill down a bit on where you were going there
Yeah.
uh, in terms of the future and how you scale this and bring it to meet all that unmet, medical burden. Um, Something Deepak Srivastava shared with us a few episodes back has really stayed with me. We're talking about this amazing progress that's been made with gene therapies for hematologic malignancy, with sickle cell being, the most tangible use case. And he highlighted this sobering reality that hadn't occurred to me because, of course, I'm so focused on proof of concept. And then, it seems like we won the race. But the current climate with the costs and the lack of a sustainable pay structure, threatening to like seriously sideline these treatments, it seems, at least at scale, for the foreseeable future. A scary idea, with the pace of this being so, breakneck. You thought it'd be a matter of years before we think of traditional therapies as a thing of the past. But I don't know. It seemed like you have a more optimistic view as you were getting at there. Do you share these concerns and or do you feel, as I think you were getting at, that there's a pathway forward to like wholesale delivery of these CRISPR or kind of CRISPR related targeting of malignancy?
Well, I share Deepak's concerns. I think that there are lots of challenges. There's challenges from reimbursement and actually being able to get, viable models for getting support for these kinds of therapies to be paid for. I think even more fundamental than that, I think a lesson that's come out of the experience with Casgevy that's been reported in a number of venues, including a recent Stat News article by Jason Mast that highlighted this, was the fact that even after approval in late 2023 of Casgevy, there's only been about 60 patients who've been able to receive the therapy to date. There are many, many more patients, even in our own clinical practice, who could certainly benefit from this. And I think one of the biggest challenges in the field has been not even the sort of theoretical aspect of how do you edit, how do you modify genomes, but really even just very basically, how do you collect a sufficient number of stem cells and then modify those stem cells in a manner to then be able to deliver those back to patients. And so there's a lot we don't know, but I think I would just emphasize to the listeners, there's still many basic challenges that often can seem, like maybe not, key challenges, but that are key aspects to how we can actually deliver these therapies. For instance, how do we best collect stem cells? How do we mobilize stem cells? That's even an area that still could be advanced. How do we expand stem cells? That's an area we've been thinking about, but certainly I think in the field, that's been limiting a lot of these therapies. And that's not just the case for Casgevy. I think it's really true for almost all the gene therapy approaches that are being applied, but even more so, a big problem in hematopoietic stem cell transplantation writ large. It's a problem for donor availability for allogeneic transplantation. So certainly, I think limits a lot of these approaches in their application broadly. And then I think even more so, currently we have to give patients basically chemotherapy that wipes out all of their hematopoietic system, but at the same time actually has risks of infertility, has risks of secondary malignancies, and so forth. And so I think that there's challenges that need to be overcome that are being, addressed, but I think would need to be overcome to hopefully make this a more widespread therapy. So I tend to be an optimist. I view there being a really bright future for this, not just for diseases like sickle cell disease or thalassemia, but happy to chat about how I view this hopefully as being a larger approach that we can apply to a range of other diseases. And this is an area that we're really excited about pursuing in the coming years. So I think that there's a lot to be done there, but I do think we need to overcome these challenges. And of course, as you brought up, reimbursement for these therapies, which are quite costly, is challenging. Getting business models that would enable the appropriate, reimbursements and, make this, cost effective for companies to produce and enable, I think becomes another challenge. So there's lots to be done, but I view it as our patients really need these therapies. And so I think given that need and given their effectiveness and given what I've seen from patients who've received these therapies and the benefits that they have, even beyond just the simple clinical data, I think really emphasizes the need for this.
Yeah, really well said. And you mentioned one of the big technical challenges, which is HSC expansion, and even now that remains to be an issue. And in fact, you've done a decent amount of work on this exact topic. Like for example, one of your recent papers in Nature Cell Biology actually showed that for apoptosis, right, could be a major driver HSC loss during ex vivo culture, and blocking it might actually improve HSC expansion, right? That's pretty cool. So, can you walk us through how you arrived at that discovery and what implications might it have for stuff we're talking about, like stem cell transplantation, gene editing workflows, or the scalability of future cell therapies?
Yeah, no, absolutely. So, that work really came from a set of serendipitous or observations that we made. So, we were several years ago very excited about studying a rare form of bone marrow failure that we had identified, and a number of other groups, including Christoph Klein's group and Fauzan Al-Kouria's group, had at the same time identified mutations in the gene called MYSM1. It was a histone deubiquinase, but in patients with biallelic mutations in this histone deubiquinase, they actually progressively lose their blood stem cells over time. They get aplastic anemia with age. And so it was a really interesting observation. But what really fascinated us, and specifically Jiawei Zhao, who at the time was a postdoctoral fellow in our group, was the idea of whether we could understand something fundamental about how stem cell regulation was occurring through the vantage point of these rare mutations. And so as he started to study this and model this in primary human hematopoietic stem cells, what he found was that the cells were being lost, but not through a conventional form of cell death, but rather through this form of cell death that is known as ferroptosis. And this is really an iron-mediated, non-apoptotic, metabolically regulated form of cell death. And it actually only been described about 10 years ago or so, or a little bit over that. And so this caught our attention. And at the time, we had really been focused on ways we could block ferroptosis to rescue the phenotype in MYSM1 deficiency. We also showed in some other bone marrow failure syndromes like Fanconi anemia that stem cell loss could actually be blocked by preventing ferroptosis. And so that was exciting. But I think also kind of left us at a little bit of a standstill, because how you could best apply this was really not clear. It was potentially helpful in these conditions, but you also worry that sometimes there's clonal hematopoiesis emerging, sometimes there's pre-malignant conditions, sometimes there's genomic changes. And so by rescuing those stem cells, you might not be doing something helpful for the patients. But as we started to think about this problem, I was really fortunate because I had another postdoctoral fellow who joined our group, Lucrezia Della Volpe, and Lucrezia got fascinated by the idea that maybe we could actually block ferroptosis and better preserve stem cells. And what she realized is she had a background in gene therapy and gene editing approaches from some of her PhD work. And she realized that maybe there was a possibility in the context of gene therapy or gene editing approaches to actually apply this kind of approach. And as we were testing this, what we intentionally wanted to try to do is actually ask, could we block ferroptosis? But could we do this across almost any culture condition, almost any stem cell source? And would this work? And that's where it got really exciting because, we started to test this in standard serum-free culture conditions. We were able to see over about a 2-week period that there was about a 4-fold expansion in the preservation of stem cells. We could see this irrespective of whether we were using cord blood as a stem cell source or if we were using adult mobilized peripheral blood as a stem cell source. So across both of these sources, we were seeing beneficial effects. But of course, there's been lots of advances that have been happening in parallel. So, for instance, there have been these great advances from Hiro Nakauchi's group showing that we could actually apply, these chemically defined cultures initially to expand and maintain mouse stem cells, and more recently to actually enable at least longer term cultures of human hematopoietic stem cells. And so we found even adding ferroptosis inhibition to these chemically defined cytokine-free cultures actually enabled even more expansion than what you would see otherwise. And so this is somewhat of an example where I think, a little bit of serendipitous observation by just studying pathogenic mechanisms led us to realize that ferroptosis was this vulnerability in blood stem cells that was actually something we could actually co-opt even for healthy stem cells and take advantage of in the context of ongoing methods development for stem cell expansion. So I'm quite excited to see that. But what I would even just more say than that is I really think this has been a big limitation in application of a lot of these gene therapies. And I'll just give you one example of this. So I had an opportunity to meet Victoria Gray, who was patient 1 in the trial of Casgevy. So she was the first patient to receive a genome-edited therapy. And in talking to her, it was really interesting because when I was asking her what her biggest challenge was, it was actually having to come into the hospital for 2 mobilization cycles just to collect enough stem cells. So she had to be in the hospital for about 6 days in total just to collect enough stem cells for the product to be manufactured where it was gene edited and redelivered to her. So I give that as just an example, but I think it's a huge need to be able to better expand stem cells. And so I think while a lot of this is at the level of how do we do this? How do we fundamentally understand some of the processes? I am hopeful that in the coming years, this is going to make a big benefit for patients.
Hmm. Yeah, what I hear from you over and over is you talk about how it begins with these serendipitous findings. When I'm like, I'm a little skeptical because it feels like when you look at as many things as you look at, that of course
Hmm.
you're going to see some things. So it's not exactly serendipity as just
Yeah.
keeping your eyes peeled. You might argue, because you've been very productive. Let's say, I mean, you've been a force. Your lab and your group has been a force in leveraging a lot of tools in the space. But I would say arguably the single cell omics approaches in large part have enabled your interrogation of all these physiological, pathological mechanisms in hematopoiesis. Um, and so I was thinking about that, uh, but thinking about, how of course the tech is a big part of that. The
Hmm.
hematology has long been just a single cell field, right? Unlike solid tissues and organs where the single cell and spatial omics has really enabled this understanding of heterogeneity right? The application in hematopoiesis is kind of different
Hmm.
because it enables a whole other dimension of analysis. And here I'm talking really about like the lineage relationships, the clonal heterogeneity, the overlay of this temporal tracing. Um, it's blown open the field. All kinds of principles, I think, have been had nuance added to them by the advent of the single cell tech. So I ask you, as someone who's at the front,
Hmm.
what do you think is the most exciting way these omics tech uh, is being applied at the leading edge, and to what end? Like, what's the thing you're most excited about in your toolkit?
Yeah, no, it's such a really great question, and of course, it's often hard to know, and hard to predict where the future will go. I do think, and I would just sort of make one comment to what you said, which is, I think in some ways, hematology and the study of hematopoiesis has been at the forefront. I think in part because, we have a stem cell population that we can do functional assays with, you know, prospectively isolating them, transplanting them. But I think even, being in the field for a little over 20 years, I've seen things evolve a lot. And I say that, I talked about when I was a graduate student seeing, these mouse knockouts and doing transplantation assays. But we had a limited set of markers that we could use to prospectively isolate these cells. And I think one of the things that's come out of a lot of the single cell genomic work from the early days, whether from single cell transcriptomic profiling, single cell epigenomic profiling, and even now some of the multi-omic approaches, is the fact that there is tremendous heterogeneity even within this population that we thought was truly functional. And so I think one thing I would just add, though, is that sometimes it's easy to forget the importance of the functional assays. And in some ways, that's sort of given us a hybrid model because, not every cell that you profile is as equivalently transplantable. And
Hmm.
yet, even within that equivalently transplantable population, we've learned that there is tremendous heterogeneity in terms of cell behaviors, in terms of the lineage output, in terms of what the fate of these cells might be, and so forth. So I think there's still a lot more to learn. But on the other hand, I'm really, really excited about the idea that now we can either prospectively label cells using things like, different types of evolving barcodes, including things like, lentiviral barcoding, but even more so endogenous barcodes, either using CRISPR-mediated genome editing. Most recently, there's been the clever use of prime editing to track these different cell lineages. So I think that there's a lot to be done there, particularly in the setting of model systems.
Mm.
On the other hand, there are lots of limitations to actually being able to apply this in human cells where you can't necessarily, modify the cells as you desire. But on the other hand, I think that there's been lots of advances in how we can use either different types of naturally occurring genetic variation that's accumulating in cells, or even other types of marks like, epimutations that occur because of DNA methylation changes at specific sites, or other types of changes. So I think that there's a lot to be done there. I think one of the biggest challenges right now in the field is how do you actually accurately detect these types of changes? How do you monitor them? And how do you ultimately get as much information as you can from those cells? And I think it's been really exciting to see, even in the past 10 years, all the advances that have happened. And I think we'll continue to see lots of advances there. And I think it'll also start to redefine, what are the contributions of stem cells to different populations? How do we better understand this process? And I think we're seeing that with studies in mouse systems, but I think it'll also certainly happen in the study of human biology as well.
Mm.
And I think you're at a perfect place to support all this translational and innovative basic science research here, in part at the Harvard Stem Cell Institute. And of course, you've got other affiliations as well. But I wanted to highlight the HSCI just because we've had a number of folks from the HSCI here on the show. It's really one of the world leading, if not the world leading institute for stem cell biology, period. And I know investigators are spread across all different programs and departments and institutes at the Harvard ecosystem. But I just wanted to highlight that, in part because I'm also an HSCI HIP alum from back in the day. So I had to shout that out in there as well. But your HCI is awesome because you're bringing all these things like developmental biology, genetics, engineering, translational medicine, all kind of under a single umbrella. And you, of course, have been embedded in that ecosystem for years now. So how has the HSCI, facilitated or shaped your work? And what do you think makes it such a like a unique environment and a productive environment for stem cell science and collaboration as well?
Well, I've really felt fortunate to be part of the HSCI. I think it's an incredible environment. As you were saying, it brings together people in very disparate areas, all of whom are focused on stem cell biology, but in maybe very different ways. And I think that's really benefited us in many ways because we've often gotten together and learned from people thinking about very different biological problems, but who are applying approaches or techniques where those can actually translate to what we've done. So I think being part of that community has really fueled a lot of the science we've done, in part because, you're bringing together people thinking about areas that might traditionally not fit under one umbrella, but that do come together because, you have this shared interest of thinking about stem cells. I would also, though, highlight another benefit that I've really been fortunate to, and you talked about it, Arun, which is the fact that um, one of the key missions of the HSCI was really in education. I was really fortunate because I've actually had a number of HSCI interns, some of who have actually returned to the lab. So I'll just give you a couple of examples. So one of the former graduate students in our lab was a trainee named Jorge Diego Martin Rufino. He finished his PhD with me, but he was originally a HSCI intern here in Boston. We got to know each other a little bit during that period of time. He was not in our lab, but then ended up coming to our lab and really had an important impact in applying genome engineering tools to a lot of the problems we wanted to understand. And in fact, some of the work I discussed about MSI2 was actually driven by Gaurav Agarwal, who was also a former HSCI intern in our lab originally. And then he actually ended up, after finishing some medical training at Oxford, coming back. And so just seeing the generations and the impact that it's had over time, on you, on others, has just been incredible. And so I think just seeing what the program has done, not only in terms of creating a community locally, but really enabling a lot of leaders to train, to get educated, and to see and come out of the system. And I would say, just even personally, I think I joined Stu Orkin's lab when the HCI was first forming, and so I kind of grew up in that community and have seen it flourish subsequently.
Yeah, in fact, that's how I met my mentor, Sean Wu, who, of course, you know very well from Stu's
Yeah.
lab. It was an agency I interned back in, I guess, 2011, and I'm dating myself now. But, he stopped by a lot of the same talks, and we had a lot of shared interests. And he mentioned that he was actually relocating to Stanford pretty soon. And so I decided to join his lab as a grad student. So, small world.
It is. It is.
Arun, you're still a young man, my friend.
Good
Um,
thing.
the scientific diaspora and then convergence at Harvard and all the elite institutes and how they seed great science in all places. It's a beautiful tree, that scientific tree, and how you can see the progression of science along the limbs of that tree to beat on that metaphor a little bit more. And so thanks for sharing that. And it's really encapsulated again in one lap. So that's a great rendering. And before we let you go, we got a couple of peripheral questions. The first being, if you were not doing what you're doing there at the HSCI and Boston Children's, Dana-Farber, all the other posts, what do you think you'd be doing with your life? For one, I'm voting for, I could use you in the air traffic control tower. That's what I'm pegging you for. You'd be paying attention to the details. It would give me great comfort on the runway. But your call. What do you think you'd be doing with your life?
It's hard to know. I mean, I think I sort of wandered into what I was doing in both science and medicine a little bit circuitously. I never thought that I would really even enjoy medicine the way I've enjoyed it. I continue to practice as a hematologist and really enjoy that aspect of my career. And so on the one hand, I could say, well, I might be a physician if things hadn't necessarily worked out scientifically, and that would be, a very satisfying career. But if it was none of those things, I actually think one thing that really fascinated me early on in my career, and that actually had been the major I'd potentially checked when I had gone to undergrad at the University of Pennsylvania, was actually to do theater arts. And so there's a part of me that says, well, maybe I'd be, doing that in some way. Although it was hard to envision how that could be a long-term career, but definitely was an area I was really fascinated by.
I think you'd make it work. I'd like to go to that theater. Finally, you have shaped so many careers and been the beneficiary of such great mentorship in your career. Why don't you share with us, professional or not, live a wonderful life? Tell us and tell our listeners a kernel of advice that you think has helped you along the way.
Yeah. I think one piece of advice I received early on in my career, and I think is still been very important, is to really have a panel of mentors. I think it's great because we often in our scientific work will have an advisor and a mentor who we can work closely with. But I think one thing that I've really benefited from is to have a group of mentors who help guide and shape my career. Even to this day, many of those are people I go to and just seek their advice. And I think it's important to bear in mind that they may not always give you advice that you agree with or that works with you or that's exactly what you want to do. And ultimately, you know yourself best. But I do think it's important to at least hear out what advice, what guidance people have. And, you might go in a different direction and that's okay, but at least it's helpful to have thought about what, they have suggested, what their ideas are and so forth. And that's been really valuable for me. And I think where I've maybe not made the best decisions or when I haven't at least had a group of people that I can go to as a sounding board. So I think it's always helpful. And the other thing I would say, and both of you discussed this from various experiences you had, but talking to different people, whether at conferences, at meetings, will help you identify that group of mentors, right? So I think really, increasing the opportunities you have to just encounter people, whether it's like Arun was saying, how we encountered Sean Wu or so forth. I mean, just the people that will shape your career are people that you'll just encounter randomly in different places. And so hopefully you can make that less of a random occurrence and more of a real occurrence.
Yes, I think that really emphasizes, as you guys talking about that scientific family tree thing, the reciprocity of it. I mean, all of the people that mentor you are doing it because they're filling their own ranks. I mean,
Mhm.
mentorship for me, at least, and I'm sure you share this feeling. It's a joy because you get to convey your experience to someone who might benefit from it. And I, for one, am never jealous with my mentorship. I would insist that you get a panel because God forbid I wouldn't want to be the only influence. I mean, having mentors that I've loved, I can pick some tragic flaws out of each one.
Mhm.
So yes, get your panel and have your own mind as well. Vijay, you're a great example of both the mentorship, receiving and giving, and you've had a great impact, both scientifically and I think personally on us all as scientists. So thanks for sharing your thoughts and all your science, and we'll have you back again, I hope, soon.
Thank you so much. It's been amazing to be here.
All right, you guys, that brings us to the end of our show today. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and roundup papers. If you enjoyed this episode, at least the second half of it, where the mellifluous talents of Arun Sharma were on display in contrast to the monotonous, soporific, meandering ramblings of yours truly, then you should please feel free to share with a friend, colleague, or on social media. You can also reach out to us on X at STEMCELL Podcast, Blue Sky at STEMCELLpodcast.com, or via email at info@STEMCELLpodcast.com with feedback or to suggest guests. or hosts.